Thank you very much. The second presentation is basically the title is on the environment and udder health, but this is a huge task. So we have basically now looking at the milking machine, which is part of the environment of the animal. We've looked on that. There was now this question, and it's a very relevant question in South Africa about heat stress. In other countries has got different problems, more freezing of teats, very cold circumstances. If you look at that bottom photo, I was quite interested in when I was in Switzerland not very long ago, but the main reason for culling animals at that stage was breaking of legs. And I asked them now, why is it so? Aren't the animals adapted to this situation? And apparently those of you that has been in Switzerland, it's a very, very small area. The farmers can't get more space for their animals. So what they were trying to do is then crossbreed them with dairy cattle to have a higher production. And these animals were not so good in climbing those mountains. So every country has got its own challenges. The other photos, you can see the mud, you can see the nice grass situation, or then, yes, we've got a mixture with the housing, with the camps, with the pasture. Bedding is always a challenge, whether available, how to manage it. Nutritional factors in the environment plays a huge role, and so we can go on. There are many, many factors play a role. And I selected then the pathogens in the udder to focus on them today. Now, just a very basic concept. If we want to achieve udder health, if you look at the left-hand side, the vertical situation, the longer the duration of an infection in the udder, the more problematic it becomes. Because the longer the infection, the higher the risk of udder damage, the other parenchyma of tissue, and that damage is usually permanent, so our cow won't produce what she could have right in the beginning. The second problem is if we've got bacteria that remain in the udder, the shedding of that material, the risk for the addition for the other animals in that herd. So the way to cope with that is to detect it as early as possible. The earlier we can detect it, and it's not always a treatment, it is once we detect it, we can change management things that would prevent other animals from getting that. Treatment, yes, once we get clinical, and in a few cases we would treat subclinical animals, but treatment is also a challenge these days. We know we don't have a large spectrum in South Africa of different antibiotics in our intramammary group. We know that worldwide there is a scare of resistance developing. So when we do treat, we need to treat it, do it very effectively. It's not a question of no use, but use it in a very effective way. Another way that we can help to reduce that infections is by vaccinations. We'll talk about that later. And then when we do have an animal that we cannot cure, culling needs to be done to get rid of the problem. If you look then at the horizontal line, the risk of the infection, where does it come from? The more there is in muddy situations, in normally warmer situations, if we've got overstocking of animals, all these things increase the risk. There's a lot higher concentration of the bacteria that can enter the udder. So the obvious is then the hygiene, the housing, the machine maintenance, which we've spoken about. And I think people often underestimate the value of this. Many of you may know the situation that was in the Netherlands where they had to reduce the antibiotic use in production animals by up to 80%, 80% within I think it was a 5 to 6 year period of time. And one of the major things that gave them this success, because they almost achieved that goal, was hygiene and management. So we should really not underestimate. All of that is work. All of that takes a lot of labor. But the effect of that is very, very good at the end of the day. So the risk of transmission is really in the parlor then, where we've got a risk of new infection, where we've got the animal that might be infected in the udder, and we'll talk just now about the two groups, one that's in the udder, the ones in the environment. They come together in the parlor, and the more of them that are there, the more is risk for udder health problems. So to achieve good udder health, it's not one thing. It is a combination that we have to look at constantly. It is something we need to monitor, that we can, in a very early stage, pick up problems so that we can try and really prevent animals from getting clinical sick. So if we look at the organisms, then they divided into two groups, and it is important that we know which bacteria is in our herd. It's not just a question of, I've got an infection on the cow, or she's got clinical mastitis. What is the best intramammary treatment or the best antibiotic? Unfortunately, our overall treatment success in udder health is less than 50%. And that is not good. And that includes the dry period. In the dry period, our success rate is double and even sometimes more than it is in lactation. So we should not waste that opportunity where animals are infected in lactation to treat them during the dry period. For certain of our organisms, the values vary a little bit. But for instance, for Staph aureus, our treatment success is around about 27%. So again, I just want to emphasize prevention rather than cure. But if we look now at the two groups, the host adapted, it's called like that because it's adapted to survive in the udder itself. It can survive there for long periods of time, which most of the environmental organisms cannot. But this is by no means cut dry, where the one can only survive in the environment and the other one only in the host. So of the environment can also survive, but that is where the main problem area is. So the host adapted, or also called the contagious organism, can live for a long time in the udder. And they are the ones that cause a lot of damage over time and a lot of shedding risk for other animals. Normally, the cell count goes up and down, but they can be responsible for very, very high somatic cell counts. Of the farmers that maybe had Agalactia outbreaks in their herds, I think the highest somatic cell count that we counted in an Agalactia herd was 80 million. And one would think that animal should have been slaughtered the day before yesterday. That particular animal, because of the way that the Agalactia bacteria work, was the cell count was down within 4 to 5 days, and there was virtually no damage to the udder. So, but it can go very, very high. Also, in our new systems where we can start detecting early problems, with the host adapted one, they start slowly raising in the somatic cell count, which can give us an indication problems is coming. Clinical mastitis might be at the door, which you'll see later when we talk about the environmental ones, that is not so. We normally see, if we can follow what happens in a herd, that the prevalence of those contagious organisms is the longer the cow is in the lactation, the more of these organisms we can actually isolate. So as the lactation goes on, the infection rate increases. And it is related to the prevalence. In other words, the more organisms we see, the more are present really in the udder itself. If we move over then to the organisms mainly coming on the environment, they are much shorter of duration because it's not the normal environment, the udder itself. E. coli is a very good example. They normally have lower cell count than the host-adapted ones, but they can also obviously increase. The problem with them is they do not give you a warning. Those of you that had E. coli sometimes would think, but this milk is really not watching properly. This morning the cow, they must have missed the cow because at the afternoon that cow wants to die. But it can be very, very quick in onset. So you basically from don't see any symptoms till clinical symptoms. A high incidence of these animals is normally shortly after calving, because that is the highest risk period. The immune system is very low in the cow at that stage. So not throughout lactation, but normally just after calving. And not so much related to the incidence, not so much related to the prevalence. Now, if we look at the major organisms in these two groups, the host-adapted one is the Agalactae, Strep Agalactae, the Staphylococcus aureus. Both of them we see in the country, and I'll give you figures a little bit later. Mycoplasma is in the country. We kind of play ostrich with this one because it's a very difficult one to isolate. It takes special methods. It also takes a very long period to grow. We have just up to now in 2 cases confirmed mycoplasma infection, and I really dearly hope that we don't see it in the future. If we look then down, the primary source is the udder. So that would be, if you've got a problem like that, your veterinarian will most likely tell you, please, at all costs, try and keep that positive animals together in a group, milk them last, because that is where your risk is. We have learned in South Africa also, especially during the beginning of the 2000, I think 5 roundabout, that the human can be a huge carrier. In the time where we had a lot of AIDS in the country, we isolated more of the human Staph aureus out of the cows than we did of the cattle strains. And we also saw at that stage that when we tested it for antibiotic resistance, the human strains were much more resistant against amoxicillin, which is used much more in people, where the cattle strain were much more resistant to ampicillin, cloxacillin. And then the milking machine, that is why we're so careful when we've got that, where the liners must be disinfected, the hands of the people must be disinfected, because they can carry it from animal to animal. It is spread during milking or any communal cloth or paper that goes from the one cow to the next. On the other side, if we look at the environmental ones, it's mainly our Streptococcus tuberous, and that, especially for our pasture-based farmers, this is our new kid on the block that's going to cause us some challenges. It is increasing in the pastures. First of all, I think there was a slow increase at first, I think because the pastures has been used for quite a long time, and this organism is excreted in the manure. So you overseed it only by the animal walking there. And these days, many of our pasture-based farmers are starting to use slurry, slurry is mainly used. Some of them is still dry manure, which is very good fertilizer, but we overseen then seeding the area with hubris. A little bit more about that later. Streptococcus dysgalactae is present, but we do not see high numbers of it. It doesn't seem to be chronic. but it is something to look out for, but not our major problem at the moment. Then the gram negatives coming from the environment. I'll give you figures later, but what we isolate mainly under this group is E. coli, Klebsiella. I've honestly isolated Pseudomonas in all the many years that got my grey hair on my head. I think 3 or 4 in 3 or 4 occasions, but always associated with water. And then the last organism, it is one with the identity crisis. It was called coagulase negative staphylococci because of the way we used to diagnose it. Now, after there's been gene typing, we realize not all of them are coagulase negative, so we call them now non-aureus staphylococci. And we'll talk a bit about them later. Now, the primary source is the environment, and it's mainly in the environment where the animal pick it up, but if the hygiene is not good, it can be spread in the parlor even. Now, why do we need to distinguish between these bacteria? Why is it important to know what the problem is in your herd? First of all, if we just look at somatic cell count, this is, if we look at the horizontal line, you can see the normal animal should remain low. Young animals should remain low throughout the whole lactation, which is very, very hard to achieve. But in the case of E. coli, that would be the first graph increasing there. If a cow does get E. coli and she does survive, you can see it peaks very high and then slowly the cell count comes down, and that can take a period of even up to a month or longer. Depending on the damage in the udder, the cell count can remain then again very low. If you look at the second graph, which is the blue one, that is a typical infection with Staphylococcus aureus. The cell count goes up, come down, up and down, and up and down, and so it goes because most of the time these organisms become chronic. What you will see as a farmer, you get an animal with clinical mastitis, you treat her, she seemed to be fine. 2-3 weeks later, another outbreak, and so it happens throughout lactation. The red line is now our Streptococcus hubris. You can see the cell count even higher, and that's what we typically see these days with hubris, quite a high cell count, 2, 3, even 4 million. And it also, at first we thought, because of it is an environmental organism, it won't last or survive for long in the udder. But there are over 100 different strains. We're at the moment busy looking at the strains we've got in South Africa, but definitely what we see in practice is that some of these strains do become chronic. So they can also last over a longer period than 60 days in a herd. As far as milk production is concerned, it usually also varies. Normally with E. coli, there's enormous loss in milk. Which recover depending on how severe it was. With aureus and hubris, there's a definite loss, and these loss are usually a permanent situation. They do not recover to the initial production. Now the coliforms, that is the gram negatives that we mainly get out of the soil overall. What we isolate in South Africa is also, we talked about the E. coli, the Klebsiella, Enterobacter, and Serratia. Now, of these, I think many of our farmers, and unfortunately, a day ago I saw a dead cow from E. coli. It's E. coli mastitis. It is a very, very sad situation because it's normally one of your best cows right in the beginning of lactation. Within a very short period of time, she's dead, and I'll show you a photo of one that survived, but how she did look. E. coli cannot survive long in the udder, and because of that, when it dies, it actually excretes a toxin. And that toxin is what can either kill the tissue surrounding that organism, and that is why we get the blue udder, or if it is excreted in large quantities, it can kill our cow. The Klebsiella we also isolate from time to time. It can cause blue udder, but I haven't isolated it from a blue udder case in the country yet. It can from time to time become more chronic, and the Enterobacter encerotia is more, it comes and it goes, usually spontaneous cure, a little bit increase in cell count, but normally not a big issue. As I said earlier, it can be the soil, the bedding, the manure, and even water. Don't underestimate the situation that water can play. Especially in your parlor, make sure from time to time, don't always only test the hygiene of the water, whether there's bacteria present or not, but also look for hardness and then, well, and bacteria, both of them. With the gram negatives, many of them actually shows clinical mastitis, especially your E. coli. So when it's in the udder, when the body can't cope with it, most of the time you are going to see clinical mastitis. So it won't be a situation that it is in your herd for weeks and months and you do not know about it. If you test and you pick up clinical mastitis very early, you will know about it. That's unfortunately not the same story with your gram-positives. Agalactae, NAS that we talked about just now can be multiple cases and you will not even know about it because the cell count varies. If we look at the coliforms, that is now the ones mainly from the environment, and this is what makes them problematic. The colony forming units, in other words, the number of bacteria, how fast they grow, they can grow very, very fast once they're in the udder and can peak after 5 hours. That is why an animal being okay in the morning, those of you that can monitor rumination, everything is fine. And then after 5-6 hours, you can see already that this animal is starting to show symptoms because of the endotoxin can be excreted. We had now the case of the animal that died. It was unfortunate, and maybe I should talk about it. Those of you that is using teat sealant, the product is good. The application is a high risk. And I want to plead with both farmers and veterinarians that's listening here. The dead cow that we saw here was because of unhygienic application of a teat seal. I think it should be, if you are a dairy farmer, only yourself or your manager should apply the teat sealant, nobody else, under absolutely strict hygienic conditions. So even if you put an antibiotic in, that antibiotic might not be able to kill the bacteria that you put in from the environment, the E. coli or whatever. And with a teat sealant, you seal it in there and you cause an absolutely super infection. There were 6 animals dried off with both antibiotic and teat sealants, and 3 of them died. Now already within 3 hours after the treatment, they could see that rumination was not effective anymore. And that animal, the first one, died within 24 hours. Basically, looking at that animal, the 1/4 was almost all the skin off, totally blue. And yeah, she was 7 months pregnant. So sorry, it's a little bit different from what I wanted to say, but I think while we're busy with it, very, very, very careful with the way you apply the teat sealant. All right, so if we go then back to what I was busy with, the neutrophil migration, because the multiplication of these bacteria is so fast, and if we haven't got cells there where they are multiplying, we cannot stop them. And hence the idea we had in the past by putting this little plastic into the teat cistern. What happens if we've got a large number of this lipopolyc saccharide endotoxin released? The phagocytosis of that bacteria is also not effective, and this will eventually, as I said, kill the cow. Yes, just a little bit of research done. This is done in Bristol, UK. They have a lot of problems during the dry period with E. coli. We fortunately at the moment doesn't sit with the same situation, but especially now in Western Cape, hopefully the rainy season will start quite soon. But the moment the rainy season starts, E. coli becomes a major problem for the calving of cows, and we need to look after that very, very carefully. Now, this is a photo of a cow that survived E. coli. It was unfortunately one of the Ornithopoid animals that were there at research station. Now, I know normally the farmers would collar, but one of the vets gracefully paid for her to be taken up in the hospital, cared for, and that whole area that you see there, the tissue that's lost, is tissue that become dead, necrotic, due to the endotoxin, and it basically falls off. Now, this tissue was already, I think, 2 weeks into healing process. Eventually, that whole area closed, and that cow walked out of there with basically 3 teats and a quarter that was not left much of. But I leave this a little bit on just to, again, E. coli is so easy to get into the udder. Again, the teat canals play such a huge role to prevent it. And then the application of any antibiotic, but as I said, especially then the teat sealant. Klebsiella, we don't find, it's not yet, it's not common. We do isolate it from time to time. It's not a major problem, but just be aware of it. The E. coli, as I said, is the one we're very wary about at the moment. Huge milk loss depending on the level of damage, and you see the extreme there, and obviously very poor treatment success. Here we have to be careful not to treat with a remedy that's going to kill the E. coli, because it's the moment we kill them and the endotoxins are released. So if we kill a lot at one time, we are going to simulate the situation where the endotoxins is released and will basically kill our animal. Klebsiella, just maybe something to think about. Normally overseas, where they've got much more problems with Klebsiella, they look at the hygiene score. One gets different hygiene score. If you look there on the left-hand side, a clean leg, a leg with about 10% of dirt. As I go low, you can see up to more than 30% of the bottom of the leg is covered by soil. So that would be a score from 1 to 4. And they just looked at 2 different farms, and they saw how the positive, Klebsiella then correlated. We had a score of 3 and 4, where the percentage then increased. Excuse me. Other very handy tool is the udder hygiene score, where we look at the udder because it's very closer to the teat itself. You can see there also on the pictures from score 1 to 4. Also a very good correlation found, and maybe if you just look at the graph at the bottom on the right-hand side, correlation between if it is very dirty, there was a huge increase in the Klebsiella, and this we found with other organisms, your E. coli as well. Now, if we move over then to the next group, the Staphylococci, streptococci, that also is mainly in the environment. The streptohubris, we said, is a problem. About 40 to 50% of them do become clinical. In comparison with the E. colis that we looked at, we 80-90% early in lactation does become clinical. So in this situation, it means we can miss a lot of these animals if we only look at clinical cases. And that is if you do that properly, stripping, looking at every milking and doing it really correctly, not milking on the floor, or milkers really concentrating on what they are doing. So it's easy to miss in the parlour. The duration, normally they take an acute infection to last less than 2 months. But as I say, what we found these days in practice is definitely that hubris, or some of these strains, is becoming more chronic. That lasts definitely much longer, and it seems to be more than 18%. Now, if we look then at our South African situation, and this is what we did at the milk laboratory, it is not the whole South Africa, but I think it gives us a not bad indication, at least, of what's happening here. If you look at the column on top, that is, if we were to test a herd, in that herd now, and we compared it with 2008 data to 2015 data, in the past there used to be around about 60% of the animals that had no bacteria. In the last, from 18 to 20, there was a higher percentage, almost 74% of animals with no bacteria. So if we move then over to the animals that had bacteria, how are they distributed in the country? And you'll see this table represents then what we call the major gram-positive bacteria. So the major guys that gives us trouble, and that would be your Staphylococcus aureus, it will be your Streptococcus agalactae, your Streptococcus hubris, and then the Discalactae. So of the animals we identified bacteria from, this group formed around about 17%. And if we look at the aureus over time, we can see there was a decrease in the bovine Staph aureus. They're still present, but a little bit less than 2015. The human Staph aureus that we isolate has decreased significantly. I think the reason is that people with immune depressant are under treatment. There is not so much shedding. So the possibility of our animals contracting that is much less, and we're very happy about that. The third Staph aureus that we've got problematic in the sense that not everybody, all the laboratories distinguish it from the normal Staph aureus. That is increasing, and the problem by that being, we still call it the Suet intermedius, although it is a Staph aureus. It is the difference is between the normal aureus is that it is not that pathogenic. It doesn't give a high cell count. It is not chronic. It doesn't cause a lot of damage. And by diagnosing that, some of the farmers are very meticulous and would cull this cow, and this would have actually been not needed. The Strep agalactae, we can see that is also much less, but we have to be careful because this is more of a seasonal thing. For you guys buying in new animals, please have them tested before you do that, because they might be dormant in the herd, and the moment you stress that animals by transporting them, bringing them to a new herd, that thing might really cause a huge outbreak. And those of you that had an agalacta outbreak will know this is not a lot of joy. Although you can cure most of the animals, you lose a lot of money because of high cell count and treatment cost. Hubris is the one that you can see is increasing, and that 2% doesn't look so awful, but at the end of the day, this data is both data from team ROs and pasture-based herds. If we were to take pasture-based herds alone, it is actually much more serious matter. And then discollector is, as I say, very low in the country. If we the gram-negative component of everything we isolated, we can see actually very, very little. It's 0.4%. But the problem is here, every time you get an E. coli, it is a very serious situation, as you could see there. The guy that we isolate most, and it is still on the increase, and it's an international type of situation, is the non-aureus staphylococci. You can see of all the organisms we isolate, they are now almost 77% of them. And having said that, they are quite difficult organism to work with in the sense of decision making, what to do with these animals. We did try and find out more of this animal. So the research we did, first of all, we looked at the different strains because we know there are many different strains. I think there's been over the 60 isolated now, to see if it's maybe one strain that will cause more problems. And we find, as described in most of the countries, that chronogenius, which is now known as the bovine strain, was by all means, out of the 200 we've done, 160 were chronogenius. The other two that was a bit prominent is the epidermidis and the hemolyticus. So we just, on the isolates we had, we just looked at the somatic cell count, and we find that the clinical cases, and remember these are not very big numbers, so percentage is a little bit not telling us the true story, but at least comparing the three groups with each other. The chronogenius seemed to be the one causing less high cell count and also less clinical signs, if you can compare it with the epidermidis and the hemolyticus here in the last column. And then very few of the other isolates. We tried to look in, is it mainly affecting early lactation or late lactation? And in the isolates we looked at, and these were from about, I think, 20 odd farms. It's not all the same farm. We found that as lactation progresses, we found more in the later lactation animals. We also looked in at parity, is it more in the younger animals or the older ones, and literature describes very often that first lactating cows, even in heifers, because it's mainly environmental organism. We also found first lactating and then in the older ladies from free lactation onwards. The perception is here in the herds where we did the trial, teat canals were also not very well preserved, that there was an easier entrance in the older cows than the younger animals, that it still had better teat canals to keep them out. As far as antimicrobial resistance was concerned, also on this organism, and that is the area that worries us a little bit, because normally the organism doesn't cause a very big increase in cell count. Very few of them do become clinical, but they can be a nuisance because they're so numerous in increasing your somatic cell count in the herd. If we look then at our beta-lactams, that's mainly used in intramammary remedies, and we compare the years from 18 to 20, there is definitely an increase, and we can see it is not looking good from a resistant point of view. Cefoxitin, very little at the moment. Our oxytetracyclines, the terramycin, is basically remaining constant. The cephalexins, cephalosporins, cephaloprins, slowly increasing, and then multi-drug resistance, we're happy at the moment, it's very low in this organism, but it's always problematic. So if we move over then to strep uberis, which is our new, I believe, upcoming problem, and this was done on quite a number of samples. You can see almost 350,000. Of that 16 or 17% of major gram positives, the uterus is almost now halfway, even more than the Staph aureus we isolate. So they are definitely something to think about. Of them in this particular trial, and that is what's worrying, they often have very, very high cell counts. You can see a large percentage of them has cell counts. Now, this is just a report of a particular farmer where we normally or regularly test the herd. This particular cow, 11170, you can see in 4 occasions of the same year where we took samples, we isolated the uterus. The cell count here in all of them, you can see from 2 million upwards. Here at the bottom, it can go, although we very seldom see an animal with uterus for 17 million. It's more of the 2-3 million. That can definitely have an effect if you've got a lot of them in the herd on your bulk somatic cell count. What we've done consequently in this particular herd, we are busy, we have isolated these uterus, which we have kept these isolates, where we basically took samples on a monthly basis in that herd, where we had a sample of the same cow every month. We kept that isolates and we're busy with looking at the genes to make sure whether it is actually the exact same strain, the exact same bacteria that lasted for that 4, 5, 6 months. We haven't completed that work, but we have already got results that there are definitely 15% or more of these organisms that remain in the udder for 5, 6 months. So some of these strains, and it seemed to be particular strains, has become chronic. Resistance here, we can see overall less than with the CNS or the NAS, but what is worrying if we look at the bottom line at multiple resistance. If you look in 2018, 5%, which was already not good, 3% in 19, and the latest one, yes, it's less data than we did with the other, but it is really worrying that 11% of them is resistant to more than 3 antibiotic groups. So it just emphasizes how careful we have to be with the use of antibiotic. When you do use them effectively, and not just 1 or 2 treatments, think the animal is fine, stop the treatment, and all you're doing is actually helping the bacteria to build up a resistance. And the next time you treat, it's not going to work the same antibiotic. All right, now for you veterinarians, you come into a herd with a problem. How do you handle it? Now, first of all, look at the green table that is there. You can have then, first of all, new infections, and that gives you an indication of where it should be. Is your new infection, the best herds would be under 5%. If new infection rate is at 8%, it's acceptable, but the moment it goes above 9%, it's not. And so forth with chronic infection, and then infection in fresh animals, that would be infection rate in the animals within 30 days after calving. Now, if we move then over to this structure, if we look at the left-hand side of the flow diagram, that is looking at the animals early in lactation. So the first question we ask ourselves, sorry, that's not early lactation, the first question we ask ourselves is a lot of the herd infected or not? If there are only a few of the animals infected, we go to the column on the left. And we need to go and see what is the problem with this particular animals, because It doesn't seem to be a contagious thing. It seemed to be more a problem with the individual animals. So they need to be segregated immediately. First of all, I identified what the problem is. If we can treat them, treat them. If they late in lactation, dry up, if they can't be treated cold, and so forth. So that is the modus operandi you would use. On the other side, what is most of the time the problem is that more than 2% of the herd is involved. The first would be then look at new infections. If there is a lot of new infections in that herd, one should ask yourself, where do they come from? So you need to go and find out, are you dealing with a contagious organism? Are you dealing with animals that's being brought in? Are you dealing with an environment that increased the risk enormously lately? Is it young animals? Is it old animals? So that is the road you will follow to find out where the source is. If there is not a lot of new infections, although there is a high infection rate in the herd, the other option is it is animals that doesn't get cured. They are infected for a long time. We cannot treat them successfully. Again, we look, is it older animals? Is it maybe heifers that come into the parlor already infected? We had work that we did where calves are fed raw milk. This was in a herd that had Staphylococcus aureus. They weren't selecting the milk for the calves, but they were present. In other words, not mastitis milk. The calves were fed there and they were kept in groups. They could suckle each other. And in this particular farm, most of the animals calving, the heifers, up to I think it was 65% of them were already infected, not necessarily with aureus, but a similar type of infection. Or is it then with older animals? You've got an aureus herd where you've got chronic infections, and the older guys is keeping that infection alive in the herd. That would be within lactation. If we've got a problem with fresh animals, the first place we're going to look obviously is in late lactation and the dry period. So again, going through that whole operation, looking the cure in the dry period, is that fine? Is there maybe in the dry period new infections a lot? So is the risk in the environment? What is the problem? And this is just a flow diagram to give you some indication where and how to go and look for the problem. So what we want to do really is avoid disease as much as possible, boost the immunity of the animal, the natural immunity, and lower the pressure of infection and things that can damage. So optimize management. This is the hard work part, but it is really very, very important. Nutrition, obviously, very important, not only protein, energy balance, but right down to trace element level. The hygiene where the animals are kept, very careful, overstocking, the husbandry, very important. Vaccination, I think there's a slide that we will talk briefly on that. Health monitoring, and I want to emphasize health monitoring, not sickness monitoring, but health. With the new programs we've got, Cow Manager that's on the market, it enables us to see when things are starting to go wrong in the healthy animal, where we can actually then proactively intervene and stop the animal from becoming sick. And then integrate a disease program, not only focusing on one, but looking at the whole situation. So I see time is catching up with us, so it is basically just the barns, the pasture, very important, the calving area, make sure that it is hygienically. Milking routine, we've briefly talked about that. Dry cow therapy, we talked about the internal teat sealant, which can be helpful if applied correctly. Vaccinations, we've got now 3 in the country. The StartVac, that's against the Staph aureus, non-aureus staphylococci plus E. coli coliform from Hipra. The one from Zoetis, that's also against E. coli and the coliforms. And now since the beginning of, I think, last year, the one against hubris, the UVAC vaccine also from Hipra. Both of the 2 Hipra vaccine works on the basis of inhibiting biofilm production. And biofilm is actually a way in which the bacteria can defend itself both against antibiotics and also the immune system, the cellular immune system of the cow. So the modus in which these vaccines work, it prevents those organisms from forming the biofilm, which makes it much more vulnerable to treatment and the immune system of the cow. I'm not going to spend a lot of time on it. We spoke about the slurry that's now being used, especially if the slurry is not heated, which we do not want these days to prevent loss in nitrogen. The bacteria survives, and then we overseed it in the pastures. The highest risk for new infections remain just after drying up, just before calving, when that teat canal is forced open. So very careful during this period. Overall, the animals should actually be treated this way. A dry cow, a conditioned horse should be correctly, it limits stress, hygiene, and I can't overestimate that. I know we cannot control the environment, but we can try to do the best we can to put the animals that are dry in the cleanest, not wet can that we do have. Maintain the udder health, and then obviously the ration remains always important. Trace minerals, very important, just maybe a little bit snipper out of this incredible, interesting, but also difficult field to understand. How to basically kill off the bacteria once it comes into the cell. Selenium, very important to basically try and get the white blood cells to the bacteria. If you have got deficiencies, that reaction time is also much less, that we talked about earlier. Copper plays a very good role in the way that the white blood cells can actually phagocytize the bacteria. If shortage, a lot of them it's much a slower process, much a less efficient process. And then your copper and your zinc plays a big role, what is going on in the cell. Not only does the cell phagocytize this bacteria, but it must kill it through basically a process we call it a respiratory burst. Peroxidized radicals are being excreted, which is then toxic for the pathogen within the cell to kill it. But on the other hand, it must be under controlled circumstances. Otherwise, it can kill the cell itself. So basically, to summarize it, selenium, copper, and zinc, very, very important in the way that your white blood cells is going to react in their ability to kill off the bacteria that comes in. So in what is important, and that what we're learning these days, within the different species, there are strain variation. Because we can do genetics, it's still a very, very expensive process, but at least we can learn what is going on in the country. That helps us in the management. Mammary health, we need to look at the risk of new infections, how long these infections are in the udder, in other words, chronic infection, and then deal with the risk of new infections. And yeah, hopefully we can create a good environment for the animals. And then by doing so, also get them like the previous animal, that they can have a long, healthy life, but then supplying the farmer over that period of time with a lot of good quality milk. Thank you very much.